Good evening, Vanchika. So from this week onwards, we'll be dealing with huge number of questions. Like I have prepared some more than 100 questions for each chapter, comprising of the previous year NEET questions. Okay, so we'll have a lot of questions to discuss. Okay, so I'm Rahul Saha, and today we'll be talking about the latter part of the chapter organisms and populations. So last time we had discussed about the various details regarding the ephemerals, the succulents, the non-succulents, perennials, okay, also the pedogenesis. Okay, good evening, Ayman. So today we'll be discussing the types of plants or soil nature and distribution of vegetation to begin with. Soil nature and distribution of vegetation. So there are types of soils like sandy, loamy, clayey, and every type of soil which have its various properties which will differ, like water retention, aeration, aeration, then mineral availability or mineral content. Okay. So that will in turn determine the nature of plants grown and also animals which could be present. Okay. So on the criterion of these characteristics of soil, the plants are divided into following ecological categories which are very important for the board examination. Okay. So plants have been divided into various ecological categories. Number one is halophytes or halophytes. You can call them halophytes only. Halophytes. Okay. These are the plants found on sandy soils. Okay. Very good objective questions these are. Then you have samophytes. Samophytes, the plants found on rocky surface. Then you have chasmophytes. Which are the plants found in rock crevices. Then you have cryptophytes. Okay. Which are the plants growing below the soil. Growing below the soil. By bulbs, rhizome, etc. Then fifth one you could be having cryophytes, plants growing on ice. Plants growing on ice. Okay. Then you have the sixth number could be oxylophytes. They are also known as calcifuges. So the plants found on acidic soils, you can say. Like maize, barley, potato. Okay. Common examples: barley, potato. Then you have calciphytes, which are the plants growing on calcium-rich soil. Calcium-rich soil. Okay. Like euphorbia. Okay. And now the next topic is, we all know that the minerals of soil are dissolved in soil water, and that soil water will be called soil solution. So soil solution is what? It is nothing but solute plus solvent. Here the solute is, you can call the solute as various minerals, and solvent is water. So you have got soil solution. So on the basis of mineral matter present in the soil solution, okay, means how much mineral content is present in the soil solution, how much molarity or how much molality or normality it has. Okay, the soils can be divided into two categories. Okay, you have oligotrophic soils or oligotrophic soil, and you have eutrophic soil. So oligotrophic means the oligotrophic soils would be comprising of suboptimum concentration of minerals. Near optimum concentration, you can call it, suboptimum concentration of minerals in the soil water. Okay, and eutrophic means it will be almost optimum, not suboptimum, means very precisely near to the optimum range or near the optimum range. So it will be almost optimum. It is near, but it is extremely near, you can say, almost optimum. Suboptimum means it could be little bit, you can say, higher or lower the range, but almost optimum means in decimal ranges. You can say, suppose if it is 7 molar, it would be suppose 6.8 molar. Okay, that would be the range. So suboptimal concentration of minerals. That is the meaning of eutrophic soil. Okay. So the total amount of water which a soil can retain per unit of its dry weight, the total amount of water that or which a soil can retain per unit of its dry weight after the gravitation percolation has stopped. The gravity would take away water via the percolating effect. So after that has ceased, gravitational percolation has ceased, is known as the water holding capacity or field capacity. Water holding slash field capacity of soil. Field capacity of soil. Okay. So it is represented as the ratio of, you can say, maximum amount of water retained per maximum amount of water retained per 100 gram of soil. Okay, that is how it is represented. Okay, so it is only 5% of the dry weight in sandy soils. In case of sandy soils, it is only 5% of the dry weight. In sandy soil, okay, maybe 15 to 25% in sandy loam. Okay. Then 20 or 35% in case of typical loam, typical loamy soil. Then 45% or up to 45% in case of clay soils or clayey soil. Then we have something known as organic matter. So the organic matter we know is added by what? It is added by the detrivorous, which we learnt in ecology chapter, by the decomposition of plants and animal remains, and also the decomposition of the excretory products of animals. Okay, so organic matter is found up to or up till 20 to 30 centimeters below the surface of the soil. And organic matter, if you want to classify organic matter which is present in the soil, okay, so number one could be litter. Litter is nothing but undecomposed organic matter. It's just excreta, you can call it. Undecomposed organic matter. So this is the excreta and also the, you can say, dead plant material fallen on the floor. Number two is duff. DUFF. That is the partially decomposed organic matter. Decomposed organic matter. It is formed of dead plant material, not completely dead. You can say some saprophytic action has happened. Okay, some saprophytic action has occurred. Okay, you can say the action perpetuated by bacteria like gram-negative bacteria like Actinomycetes, fungi and other bacteria. And fungi. So they have released their enzymes and till a level decomposition has occurred via hydrolytic enzymes released by various microorganisms. Then in the end, the last part or the final stage is formation of humus. So it is the fully decomposed product. Decomposed organic matter. Okay. And it is a you can say finely divided, dark and amorphous too. Dark and amorphous organic matter too. Okay. So it contains carbohydrates, sugars or sugar alcohol, lipids like fats, oils, waxes, tannins, resins, lignin. It contains many stuff. Okay. Tannins, lignin. Okay. Then amino acid. very very nutritive. Okay, in purines, pyrimidines, alkaloids, carotenoids, etc. You can say it contains 55 to 60% of carbon. Okay, 35 to 40% oxygen, 35 to 40% oxygen, and 5% hydrogen and 5% nitrogen. So that's the content of humus. Okay, and the formation of humus is called humification. And later on, I had explained to you in the ecology chapter only that the breaking of humus down into minerals, carbon dioxide, and water is known as mineralization. So organic matter is a tremendously vital source of minerals to the plants. It also increases the water holding capacity, the porosity and aeration of the soil. Okay. It also contains a certain acid known as humic acid, which increases soil fertility also. Humic acid for soil fertility. Okay. So this is all about the soil that can be asked in your board examination with regards to organisms and populations. Okay. Now the next topic is, how do organisms cope up with environmental changes? Something is known as, in the commencement or during the commencement of the chapter, I had told you about physiological ecology. Physiological ecology. So this refers to adaptations of different organisms to their environment. How are you biochemically adapting? How are you epigenetically causing silencing or upregulation or downregulation of any gene? How are you causing, you can say, variation acquisition? Are you causing frame shift? Are you causing insertion deletion? Are you skipping some nucleotides during the replication process that this deletion is happening? Are you over replicating, okay, by forming loop, thereby insertion is happening? Are you going for trade-off, thereby reducing the size of one chromosome and increasing the size of another chromosome, which is again known as reciprocal translocation, which may become detrimental if you go for formation of chromosomes like Philadelphia chromosome 9 and 22, which I had explained to you. So are you going for all that stuff? That is adaptations of different organisms of different organisms to their environment. Okay. So we all know, before understanding it, we all know for a fact that environment and the person or the species or the individuals of the species living or thriving in that specific habitat are all correlated, interrelated. So we shall never take the abiotic components of the environment to be static. So many people, they make this tremendous or drastic mistake, or it's a flaw, but abiotic components are never static. Okay, one can chop down the trees, one can cause glaciers to melt, our actions, we are driving, you can say, polythenes and excreta without, you can say, HTP, right? Treatment. So what we are doing is, we are increasing the biochemical oxygen demand, we are decreasing the dissolved oxygen. Okay, sewage treatment plant is not into action, so we are changing the abiotic conditions, abiotic factors in different, you can say, hydrosphere, lithosphere, atmosphere, by excreting or by secreting many gases, sulfur dioxide, trioxide, chlorofluorocarbon into the air. So we are always changing. It's an, you can say, it's kind of a dynamic process. You can say it's not a static process, okay, at all, if you think. So one changes during the time of ecological succession that I had taught you, nudation, and then pioneer species. Okay, so I had also taught you that the species also have an effect on the environment, and the environment also changes. Okay, then you have serial communities, and in the end you have climax community. Okay, so there are various morphological characteristics which will change in an individual, but also in the abiotic status when it comes to the abiotic status of that specific habitat. Okay, so in living organism the homeostasis is maintained by acquiring adaptations by morphological, in, you can say, biotic or living organisms. The homeostasis is maintained by acquiring variations or changes in the morphological, physiological or behavior, by acquiring changes in morphology, physiology or behavior, or morphological, physiological or behavioral patterns. Okay. So this is how one can adapt, and if you do not adapt, natural selection law will be applied on you. It is always applied on you, and then it will cause eradication of that species. Okay, unfit individuals. Then if you are able to adapt, you will be having comparative reproductive success, and you will be also having differential reproducibility. Okay, so that is again neo-Darwinism. So these are all very, very interrelated, right? Darwinism, neo-Darwinism, evolution, ecology, populations, these are all interrelated, one after the other. Okay, good evening Ayushi and Tarun. So these are all interrelated. So the thing is, number one which comes in the board examination is regulate. Some organisms are regulators. Regulation involves thermoregulation and osmoregulation. Thermoregulation and osmoregulation. So thermoregulation is what? It is keeping the body temperature constant. Okay, means it is able to adjust the body temperature on its own. Internally we can keep the body temperature constant. Internally we can adjust the body temperature, not depending or not relying on the outside temperature or external body, or you can say the temperature of the system which is outside of the body. Okay, so that is known as, those kinds of organisms are known as homeothermal or warm-blooded creatures. Okay, so they can also be known as endothermic creatures. So they can regulate their own body temperature. Okay, even if you go to Arctic, even if you go to Sahara, you'll be able to regulate your own body temperature till an extent, right? You have the thyroxine hormone, the tetrodotoxin, you have the triiodothyronin, the T3, which is more of an active form. Okay, produced when TSH binds to the thyroid glands, the thyroglobulin, the tyrosine, the iodination of tyrosine, 3 iodinations, 4 iodinations, those will happen. Okay, T3, T4. So what happens is 3 tyrosines and 3 iodination is T3, and 4 tyrosines and 4 iodination, each iodine connected to one tyrosine residue is the T4. What happens is, I have already explained that tyrosine induces this NAK ATPase pumping, or it causes the expression of it, and Na plus is less inside, Na plus is more inside, K plus is less extracellularly, K plus is more intracellularly. So it still causes 3Na plus content or 3Na plus to go out, 2K plus to come in against their concentration gradients. So this is going against the slope, so it will require ATP hydrolysis. This is an example of active transport energy. This energy will be used to pass this transport against the respective concentration gradients of the ions through the ion channel NaK ATPase. It is known as Na plus K plus ATPase channel. Okay. ATPase pump, which you will learn in higher classes, it's a type of P-type ATPase. There are multiple types of ATPases. Okay. Now, some of the energy is used to increase the body heat also, that we can survive in the, you can say, Arctic region for few hours, or you can say half an hour before hitting or before being a victim of hypothermia. Same way we sweat and evaporation causes cooling. So that is how we can survive in the Sahara Desert before being a victim of heat stroke. But we can survive for a few or you can say for a few hours, right? Then coming to osmoregulation. So osmoregularity is maintained via solenocytes or flame cells or protonephrodia in case of platyhelminths. In case of platyhelminths like planaria, acylomate, triploblastic creatures. Then in all vertebrates we have kidneys, right? We have nephridia in case of, you can say, annelids. Okay, kidneys in case of vertebrates. And then we regulate ultrafiltration via glomerular capillaries. We have antidiuretic hormone, we have aldosterone, right? So all these things come into picture. So if I am teaching you the kidney chapter, you must have learned kidney chapter, okay? So you might have learned for a fact that the osmolarity of human blood is nothing but 300, okay, 300 milliosmol per liter, the osmolarity of human blood. They are countercurrent exchange and countercurrent multiplication. Exchange via vasa recta and multiplication via the thick ascending limb of loop of Henle comes into action. Okay, 300 milliosmol per liter is fixed. Okay, so if it increases, suppose I am dehydrated completely, or you are dehydrated, you have gone for a games activity period, or just gaming, running, strenuous activity. So you have literally lost a lot of water via perspiration or sweating. Then every one of you might have noticed at some point of time in your life, almost on a monthly basis, like not during quarantine, but when you are actively going outside. So you might have noticed that the urine turns quite concentrated after you have gone through a period of excessive sweating or perspiration. So urine volume also becomes less. You urinate lesser volume of urine and it becomes yellowish, right? It's not whitish anymore. So why is that? Because the body does not want to lose any more water. Because if it loses any more water, what will happen is the blood will become more and more viscous, which will increase the event of or likelihood of having a myocardial infarction or heart attack. Because the heart, it is pumping inotropic effect, but it is not able to pump, or it won't be able to pump a very viscous like honey like blood. Okay, so it may not reach all the organs, and you can have hypoxia induced free radical damage in various organs, leading to non-availability of HbO2 because blood is too thick, right? And then ATP deprivation and cell death or necrosis. So in order for that to not happen, in order to prevent that specific event, you have something known as hypothalamus. Hypothalamus releases vasopressin, presses the vessels, okay, causes vasoconstriction, also known as antidiuretic. Diuresis means urine, more and more urination, more and more water loss, means antidiuretic. It will prevent diuresis or water loss, antidiuretic hormone. Okay, so it is secreted by hypothalamus, stored in the posterior pituitary gland, or also known as neurohypophysis. Okay, and when the neurons known as osmoreceptors, these are the neurons, they can sense the osmolality of the blood. Where suppose the osmolality has reached to 350, so they will cause the hypothalamus to signal the neurohypophysis to release vasopressin. This vasopressin will go and bind to the distal convoluted tubule, DCT, and cause expression of aquaporin 2 channels on the DCT cells. And aquaporin are the channels which will cause reabsorption of water. It will cause more and more reabsorption of water. Okay, so in this distal convoluted tubule, water will be reabsorbed, okay, through aquaporin channel expression caused by vasopressin, and then you will be having very dilute urine. And when you rehydrate yourself by drinking a lot of water, osmolality falls, and then that signal gets auto-regulated. That is the feedback mechanism, negative feedback, and then ADH is not secreted anymore. And then in the absence of ADH, the aquaporin 2 channel expression is lost. It is again sequestered in the endosome, okay, this channel, and now this reabsorption cannot happen, and more again urine is released, and more dilute urine, more whitish urine. So your body keeps a level. Too less osmolality is also a problem, too high osmolality is also a problem. Too much will cause increase in BP, because the lumen of the artery after vasodilation can reach a certain threshold limit. You cannot put 10 liters of blood by drinking more and more water. So too much intake of water is also poisonous. So only 1500 ml to 1800 ml of water shall be drunk a day if you are not perspirating. Okay, not 7 to 8 liters. Okay, so that's the rule. Okay, so that is known as regulators. Okay, then we have something known as aldosterone. So aldosterone is a hormone which is secreted after RAAS system, renin angiotensin aldosterone system. Again, going back to the kidney chapter, you have synthesis of renin if the GFR is too high. Okay, blood pressure is too much, GFR is too high. Normal GFR is 125, you can say, ml per minute in males, 105 in females. If GFR is too high, renin is secreted by the macular denser cells of juxtaglomerular apparatus, macular denser cells. And again, directly it does not make renin. You have pre-prorenin, then prorenin, and then renin is secreted. Renin will do what? It will convert angiotensinogen, which is secreted by the liver. Angiotensinogen, which is secreted by the liver. Renin will convert it into, it will cleave certain amino acids and convert it into angiotensin 1. Then ACE1, angiotensin converting enzyme 1, secreted by the epithelial cells of the lungs. It will convert angiotensin 1, cleave some more amino acids and convert it into angiotensin 2. And then this angiotensin 2 will go and bind to, you can say it is a vasoconstrictor, it will constrict the blood vessels, okay, lower GFR, increase blood pressure, and also will bind to the zona glomerulosa layer of the adrenal cortex, and will cause release of mineralocorticoid hormone known as aldosterone. Aldosterone will again go and bind to DCT and will cause more sodium reabsorption. More sodium reabsorption will lead to more water reabsorption via osmosis, and again more blood volume. Again it will increase BP. Okay. So both these things will increase BP. So antihypertensive or BP lowering drugs act on blocking the aldosterone receptor or angiotensin 2 receptor or ACE inhibitors, directly this ACE enzyme, or also renin inhibitors. So that's how you can treat hypertension too, okay, by recommending drugs like enalapril, okay, ramipril, which will be ACE inhibitors, all the sartan family, olmesartan, telmisartan, which are the angiotensin 2 receptor blockers, receptor antagonists. Okay. This is all about your regulation. Now we have something known as conformers. To confirm. Confirm means the poikilothermal or ectothermic or cold-blooded animals, they are not able to regulate their own body temperature. Okay, they cannot maintain a constant internal body temperature, and their temperature would invariably change with the change in the environmental temperature. So the thermoconformers regulate their body temperature in the following two ways. Number one is cyclomorphosis. The cyclomorphosis I have already taught you, that change in the body shape and size due to these seasonal changes, which I taught you in the last class only. And then you have periodic activity also, short day, long day, day neutral, plants and animals both. And then, so number one would be cyclomorphosis, which has been already taught. Then periodic activity, it has already been taught in the last class. So the third one is by maintaining large surface area to volume ratio. To volume ratio. So what happens is animals like shrews and hummingbirds have larger surface area relative to their volume. Why? Because it helps them lose their extra heat rapidly when it is cold outside. Okay, and the downside is they have to generate a lot of energy in order to gain body heat. So again, this is the main reason why such small sized animals are, you can say, rarely found in the Arctic region or in the Antarctic region, or you can say in the polar regions. Okay, so these species have evolved, you can say, for the ability to regulate their body temperature, but only over a limited range of environmental conditions, even only if they are able to live or are living persistently in hotter regions, so that will be beneficial. So if they go and dwell in the colder regions, this will be an anti-regulatory effect for them. Okay. Then you have osmoregulators also, osmoconformers also. So osmoregulatory, I have told you. Now you can say conformers are also there. Okay, these are those animals which have wide range of osmolarity. Okay, nothing happens because we are osmoregulators. So we only want 300 milliosmol per liter. Okay, but if you have a wide range of osmolarity that you can tolerate, then you are at a really advantageous point. Your osmoconformer means they can tolerate, osmoconformers, they can tolerate a wide range of osmolarity. Means they can live in fresh water also, in seawater also, in any kind of saline condition, right? So you have got a large range or a wide range of habitats open for you. Osmolarity. Okay. Then the third one which we have is known as the migrators or migrate. Third set of morphological or behavioral adaptation or physiological adaptation. Under that category we have, this is a kind of behavioral known as migrate or migrators. So it involves both shorter and longer journey. Migrators, short journey, you have long journey, both are present. Okay. So they are undertaken by the animals to resist what? To resist the temperature extremity. If they cannot resist a specific temperature, if that is too extreme for them, they'll just migrate, they'll leave that specific place and they'll depart. extremity. Okay. So what they'll do is they'll temporarily move away from the stressful conditions. Move away from the stressful conditions, okay, to a more favorable area. Okay, and return to their original habitat. And return to their original habitat when the stressful conditions are no longer present. Stressful conditions are no longer present. So this is what the migrators do. So if you talk about long trip migrators, okay, so birds like, so long journey ones. So birds like Arctic turned birds, or you can say a better example would be Siberian cranes, American golden plover. What they do is every year thousands of migratory birds living and thriving in Siberia and other extremely cold northern regions, they reach the renowned Ghana National Park in Rajasthan, okay, during winter months. Why? Because at that period of time, the Siberia becomes extremely cold. Even they cannot tolerate the Siberian climate that point of time. So they migrate to, you can say, Rajasthan, because it's winter here also. And this winter, you can say -5 to 10 degrees Celsius is tolerable. But that point of time, the Siberia can become -35, -45 degrees Celsius, which will be intolerable. That would cross the thermal extreme threshold for these birds, even though they thrive in Siberia, because they can thrive in suppose -20, so from ranging from -20 to +10. So that is their range. But if you take it down, or if you lower it much below -20, -35, -45, then they will perish and die. So then they take the route of migration. They take the option of migration, the long journey migration, and reach Rajasthan. Okay. Then certain animals like reindeer, short journey. So reindeer, okay, migrate from temperate, you can say temperate coniferous forests to tundra biome during the summer months. Okay, because there will be too hot, so they'll be traveling down the lane to tundra biome during the summer months, so are known as short distance migratory animals. Okay, from temperate coniferous region of forests to the tundra biome during the summer season. So last one is suspend. What they'll do is they'll lower their metabolic rate, their basal metabolic rate, their catabolic rate, and anabolic rate. They'll stop synthesizing or lower the synthesis of the enzymes required in their various biochemical activity, biochemical cycles, biochemical pathways. And then what they'll do is they'll try to spend as less energy as possible. Right? That is the main aim of suspension. They are going for suspending all their metabolic activities and trying to reduce the ATP consumption as less as possible, just for their survival. They do not want to indulge in metabolic activity. They just want to live that period out. Okay, let it through. That's all they want to do. They want to survive and not engage in, you can say, hunting, and you can say food eating, devouring on food and various other activities. So they just want to live it out. They want to pass that period through. That's all they wanna do at that point in time. So suspend means again they will be poikiothermal organisms, very less homeothermal, mostly poikiothermal organisms. Okay, which are not able to migrate. Okay, they cannot migrate to avoid stressful conditions. Now, what are they going to do? They have that one last option. The last option is suspend. Okay, so they can attain suspension or escape or evade such conditions by living through such conditions in one of the following ways. Number one is hibernation. Also known as winter sleep. Hibernation is also known by the name winter sleep. Okay. So you must have heard of something known as uncoupling factor. Uncoupling factor in mitochondria. So what it does is, it is an inner membrane protein, okay, which is capable of dissipating the proton gradient generated by NADH powered pumping, okay, into the intermembrane space, and can produce, you can say, energy. Okay, it can produce energy via this specific process and can break the chain of F1 F0 induced ATP synthesis, or you can say phosphorylation of ADP. So instead of making too much ADP, it can use the, in simple terms I can say, the UCP protein, uncoupling protein 1. It can cause the production, or it can use the electrochemical gradient which is present. If you have studied it properly, you will know it. Electrochemical gradient in the inner mitochondrial membrane, in the intermembrane space, it would go back into the inner mitochondrial membrane via the chemiosmotic theory. Okay, and binding change model causes the, you can say, formation of ATP. So it doesn't want to form ATP. It uses this gradient, this potential energy, to generate more and more heat. Okay, thereby the polar bears, they do not die. So they are only the you can say, homeothermal creatures that require hibernation. So other ones, like you have amphibians, you have reptiles. Okay, amphibians like frogs. So this is for polar bears. They can generate heat during the sleep period. Okay, so amphibians like frogs. You have reptiles like lizards and snakes. Okay. And you have also some insectivorous homeothermal creatures like shrews, bats, ground squirrel, okay, jumping mice. They also undergo hibernation. Okay, and polar bears of tundra biome, I have already told you, especially of tundra, Arctic and Antarctic region, they also undergo hibernation. Okay. So the next one is known as something known as estivation. Estivation is known as summer sleep. Summer sleep. So it is the period of dormancy during the summer months. Just opposite to hibernation is a period of dormancy during summer months. Okay. So as to escape from the extreme heat caused by the scorching sun. Extreme heat period. Okay. So it's commonly observed in lungfishes. Okay. The ladybird beetles, certain snails. Okay, these are the examples. Ladybird beetles, certain snails, etc. Then we have diapause. Diapause is a type of aestivation only, okay, but little bit different to aestivation. Diapause, you can call it a special type of aestivation where what happens is the morphological growth is ceased. Okay. This is a special type of aestivation where what happens, the morphological growth and development the morphological growth and development is halted, is ceased in order to avoid, you can say, various kinds of ailments or problems related to extreme heat in the summer season. It is not, you are not suspending yourself completely, you are not suspending your metabolic activity after you have achieved growth. What you are doing is you are ceasing your growth altogether. Okay, suspending complete metabolic activity and suspending also the growth. Okay, that is diapause. So growth and development is also suspended, ceased or halted. Okay, so it has been reported in many insects. Okay, then crustaceans. Then you can say mites, snails also, and zooplanktons also. Zooplanktons also. Then the next way of going for suspension is encystment. You have encystment and excystment, formation of cyst and deformation or destruction of cyst coming out of the cyst. So it is in bacteria, fungi and lower plants. What we have is we form cyst. Cyst formation is seen. You can use chitin, N-acetyl glucosamine to produce cyst in case of fungi, right? So bacteria, fungi and lower plants they produce thick-walled spores, so bacterial spores, fungal cysts. Okay, so thick-walled spores are produced. So bacterial spores are much harder than fungal cysts, but fungal cysts they do the job properly again. Okay, so they are thick-walled spores which are formed during the stressful conditions. Again, it will lower the metabolic activity to almost zero, cease the metabolic activity, and it will only burst. The cyst will only break, and the cyst is very, you can say, enduring. It is very resistant to temperature changes, to acid changes, to salt changes. You can say you can put it in hypertonic, any temperature. Okay, so it doesn't pressure, so it doesn't break, doesn't let the bacteria inside of fungi inside die, right? Stressful conditions, and it will only break once conducive conditions return. Okay, that is known as cyst formation. Okay, so bacterial cysts or bacterial spores are really the best, you can say, and highly dormant. Okay, so that is known as bacterial spore, much better than fungal spore. So it is also known as endospore in bacteria. If you want to know bacterial, the endospore in bacteria, okay, that consists of, you can say, the DNA ribosome and large amount of, you can say, dipicolinic acid, okay, and dipicolinic acid is a spore specific chemical that appears to help in the ability for endospores in order to maintain the dormancy period, okay. So it accounts for almost 10% of the spore's dry weight. So in the next class, we'll discuss about the population density and the J curve and the logistic curve, okay, and the adaptation various rules like the Allen's rules, Jordan's rules, okay, Bergman's rules. So these things will be discussed in the next class. So I hope you were able to comprehend this lecture properly, and if you have had any kind of doubts or queries, you can ask me on my personal number. I'll reply to you as soon as possible. Okay then, thanks a lot. Let's meet you in the next class.